Hi class, in this recording we're going to focus on histology. So let's dive into it. Oh, I went 2 slides there, my bad. Okay, so when we think of our body, we have lots of different cells, approximately 50 trillion cells in our body, so quite a few. And it's worth noting that there's approximately 200 different cell types. We can take those 200 cell types and break them up into 4 broad categories: epithelial, connective, nervous, and muscular. Epithelial and connective we're going to spend the most time talking about. Nervous and muscular are the short and sweet categories. Now, when we look at an organ, we define an organ, or I like to define an organ as something composed of 2 or more tissue types working together for a common purpose. Now, when we think of organs, and this is from your text, lecture text, this organ is going to be a structure that has discrete boundaries, and those boundaries are typically going to be made up of different tissue types. The inner tissue type that does the work is typically called the stroma. The outer tissue is typically going to be the membranous tissue that serves as the boundary. And that's a bit of an oversimplification, but it's worth just having that framework moving forward. When we look at histology, histology is the study of, ology for study of, histo, think of tissues. So histology is the study of tissues. It involves working on a microscope, because to look at the structures of tissues, you need to spend a lot of time working with visible light microscopes. Typically, those are the tools of choice. Occasionally, electron microscopes will be used. And as we look at the tissues in our bodies, our tissues have 2 big components. There is the living component. So as we look at that living component, that'll be the cell. And then there's the cell products. And the cell products are oftentimes referred to as the matrix. These cell products are usually going to be extracellular. And for the sake of simplicity, because they are smaller than a cell, I refer to them as the non-living component, because the cell is the smallest living thing. It's the structural and functional unit of life. As we look at the living and non-living component of a tissue, the cells and the extracellular matrix, they work together to have that specific function. Then as we look at these 4 tissue types, the epithelial, connective, nervous, and muscular, they have different structures and functions. We're going to focus on the different structures of the cells, characteristics of the matrix, and the amount of space occupied by the cell versus the amount of space occupied by that extracellular matrix. Some of these tissue types, like epithelial tissue, have almost no extracellular matrix relative to the volume of their cells. Other tissue types, like connective tissue, have comparatively a high percentage of extracellular matrix relative to the amount of volume dedicated to the cells themselves. Our extracellular matrix is going to be made up of protein and a clear gel-like substance called ground substance. Depending on the particular tissue type, sometimes the ground substance is highly viscous, sometimes it's serous or watery. This ground substance, this liquid outside of the cells, is also referred to as extracellular fluid. It can be interstitial fluid, it can be tissue gel, it can be called tissue fluid. There are multiple names for it. When referring, generally speaking for me, just so you have full disclosure, when I'm talking about a specific tissue type, I like to refer to it as ground substance. The context of talking in histology means I'll refer to it as ground substance. When I'm talking about organ systems and nutrient transfer, I typically, outside of the discussion of histology, will refer to it as extracellular fluid. So that's my personal bias as your instructor. Now, as we look at embryonic tissues, this is where all the magic begins. We all know the story of the birds and the bees. So we have a fertilized egg. That fertilized egg has cell development initiating and becomes an embryo. That embryo is then going to have layers of tissues. The layers of tissues can be broken up into 3 big layers. And these early 3 layers are referred to as germ layers, because these are the germinating. The root word here is related to germinate, thinking when you think of plants germinating in your garden bed, because these are the very early stages of tissue development. We have the ecto, endo, and mesoderms. Ecto means out, endo means in, meso means middle. So as we look at the ectoderm gives rise to the tissues on the outside of our body, notably the epidermis. One of the things that is a little bit more confusing for students is that when you look at embryology and the formation of our bodies, our nervous system starts out on the outside of our bodies. And then there's some folding over of tissues so that the nervous system ultimately ends up on the inside of our bodies. When we think of the endoderm, think of the tube that goes on the inside of your body, mouth to anus. So think of digestive tracts. When we think of mouth to anus, the glands associated with the digestive tract. Also think mouth to lungs. So the tubes that go from the outside to the inside of your body. So when we think mouth to lung, think respiratory tracts. Tract, singular. As we look at the mesoderm, this is the middle stuff, and this is going to become the mesenchyme. And it's going to have lots of collagen fibers and fibroblasts within its gel matrix. A fibroblast, blast means build. It's a cell type that builds stuff. What does this cell type build? This is a cell type that builds fibers. So a fibroblast is a fiber building cell. So we have within the mesoderm is going to have a lot of fibroblasts making fibers like collagen, and the mesoderm is going to give rise to the bone, blood, muscle, and cartilage tissues of our bodies. Now, as we look at histology, this is kind of tough. It involves a little bit of mental manipulation. When I say mental manipulation, you need to look at a two-dimensional image and convert it to a three-dimensional structure. And that's kind of tough. But before we can look at that two-dimensional image, we need to make a two-dimensional image. We're going to start with a tissue, and we're going to have to fix it. We fix the tissue to prevent it from decomposing. A common fixative is alcohol or formalin. Formaldehyde, mercifully, is not used very much anymore. I've worked with formaldehyde, and I've had to dissect animals preserved in formaldehyde because I'm a little older and crusty. And oh gosh, it was disgusting. Oh. As we make our sections, you could think a section can be referred to as a slice. And our histological sections are super duper skinny or thin slices. They're made with a special instrument called a microtome, and a good section is 1 or 2 cells thick. Earlier this spring, I ordered some dense bone microscope slides to get some more for you to work with in the face-to-face lab setting. And the slides that I ordered were awful. They were about, I want to guesstimate 10 to 20 layers of cells thick. And when you have that many layers of cells, it's really difficult to individually isolate the characteristics of one cell because there's cells above, cells below. It's all out of focus. It's a hot mess. So we first preserve the slot tissue to keep it from decomposing. Then you make a super skinny section. And then after you make your super skinny slice or section, you stain it. And these stains are going to artificially color specific parts of the tissue. So we're going to have different cellular components absorb different colors so they're easier to see. And this whole process will take that three-dimensional structure and turn it into a two-dimensional slice. And when we look at this two-dimensional slice, we are typically going to have a lot of problems. If we have a cell with a centrally located nucleus, some slices are going to miss the nucleus. Either the nucleus would have been above or below, so you just won't see it. So in some slices, the nucleus and cell are going to appear smaller just based on how it was sliced. And then if we have curved or coiled tubes or ducts, they might not be continuous when you look at them in 2 dimensions. This slide really is referencing these pictures right here. So when we think of a cell and the nucleus, I want you to think of this egg. You can think of a hard-boiled egg as a cell with a nucleus, where the yolk of the egg is the nucleus. And if we slice it right down the middle, it looks something like this. But if we slice the egg off to the side, the egg itself can look smaller. You know, green on green is really hard to see here. Give me a second. Here we go. I'll go to red. The egg itself looks smaller, and the nucleus looks smaller. Or if we go off at a very oblique slice, you can have a very small looking section of the egg and no nucleus at all. All of these things are slices of the exact same structure, but depending on where it was sliced, we have very different appearances. And let's look at tubular structures. We'll start off with something easy like a macaroni noodle. So as we look at this macaroni noodle, if we slice it right at the tip, all you get is two circles in a two-dimensional view. If you slice it at the other extreme end, you just get a little oval. And if you slice it right in the middle, you get two circles with a biconcave shape in between them. So depending on how you slice a tubular structure, you can get very different looking views. Here is a coiled duct, and it is not uncommon in our body to see coiled ducts like that. And if we slice through that coiled duct, you can end up with a structure that looks like this in 2 dimensions. So when you're looking at a microscope slide, bear in mind it's a 2-dimensional view of a 3-dimensional object. As we're looking at our sections, there's some terminology that you need to be aware of. On microscope slides, if you see an LS, lowercase ls, that's an abbreviation for longitudinal section, and the tissue is cut on its longitudinal or long axis. So if we were to cut a long bone or on the long axis, it would look like this. A coil or tubular structure would look like that. If you see CS or XS, that is an abbreviation for cross section. So if you see those letters on your microscope slide, that means we have a cross section or transverse section of the slide. So the structure was sliced perpendicular to the long axis. So I'm a really big fan, for the most part, of cross-sectional slices. Every once in a while, I like the longitudinal sections, but those are the exceptions. I prefer cross-sectional slices of most tissues. So here we have a cross-sectional slice of a long bone. Here we have a cross-sectional slice of a blood vessel. And then we have the oblique section. When I think of oblique sections on microscope slides, this is the miscellaneous category. So that's not longitudinal, it's not transverse, it's anything in between. And then you end up with more confusing views. And really on a lot of microscope slides, if it's through an entire organ, you're going to have longitudinal sections, cross sections, and oblique sections all on the same microscope slide. So it's important to have that good frame of reference to know what you're looking at and how to interpret that 2-dimensional image. Now, as we're looking at microscope slides, there's some other terminology that you need to be familiar with. A smear is when you take that tissue and you rub it across the slide. And you typically start off with a microscope slide, and then you put a drop of tissue on, oops, excuse me, I meant to go blue here. You take a little bit of the tissue, just put a blob of the tissue across it, and when I've made smears, I then would take a cover slip, I meant to go with green there. So let's take our green cover slip, and you just use the cover slip to drag the tissue across the slide. Blood tissue is typically exclusively presented as a smear. Smears are used for tissues that don't have a lot of fiber within their extracellular matrix, or tissues that are liquids, typically at room temperatures. When we work with a spread, our spread is going to be done or used for a tissue that's typically not a liquid at room temperature. So let's diagram a spread. If I have my microscope slide, I would take my chunk of tissue, put it on the microscope slide, and then I would take my cover slip and I would put my cover slip over the tissue, and I would just apply a lot of downward force, causing the tissue to spread out as opposed to being smeared like I diagrammed on the front or on the top of the screen. That's all we have for this recording. If you have any questions or comments, please feel free to shoot me an email or to post them in the class discussion board. And as always, happy studies.